The overall goal of the following experiment is to determine the percentage of cytosolic and vacuolar Salmonella typhimurium in infected bone marrow-derived macrophages. This is achieved by infecting macrophages with M-cherry-expressing Salmonella typhimurium. As a second step, cells are treated with the detergent digitonin, which permeabilizes the plasma membrane but not vacuolar membranes. Next, cells are stained with an anti-Salmonella antibody coupled to the fluorophore PHITC in order to label cytosolic Salmonella typhimurium. The results show the percentage of cytosolic bacteria among total bacteria based on fluorescence-activated cell sorting, or FACS.
These methods can help answer key questions in the field of host-pathogen interaction, such as, does an intracellular pathogen replicate in a vacuole or in the cytosol?
Inoculate the bacterial strain 1 day before the infection in 3 milliliters of Luria bertani medium supplemented with streptomycin and ampicillin in a shaker at 37 degrees Celsius overnight. To account for the individual conditions, add cover slips to the wells of a 24-well plate that will be used as controls. To plate the cells for infection, seed wild-type bone marrow-derived macrophages, or BMDMs, at a density of 0.125 million cells per well in 1 milliliter of primary macrophage medium. Then, incubate the cells overnight in an incubator at 37 degrees Celsius and 5% carbon dioxide. The next day, determine the concentration of Salmonella in the overnight culture by measuring the optical density at 600 nanometers, or OD600, of the diluted culture against phosphate-buffered saline.